Hello and welcome. In this lesson, we want to show that photosynthesis has occurred in a plant. Now, one of the most convenient way to show that photosynthesis has taken place is to test for the presence of starch in a leaf. Why is starch? Because during photosynthesis, carbohydrates are formed and excess glucose is converted into starch that is then stored in starch granules within the chloroplast. Now, how do we make sure that the starch that has been formed through photosynthesis and not from the storage regions? In other words, how do we ensure that the starch that is present in the leaf has been formed through photosynthesis and it's not as a result of carbohydrates being transported or translocated from other parts of the plant to the leaf? Now, the way to ensure that the starch is just from photosynthesis and not any other source is by discharging the plant. So we discharge, and discharging involves placing the plant, and conveniently to be a potted plant, in darkness for a period between 36 to 48 hours. And during this time, all the starch in the leaf will be hydrolyzed, and either the glucose resulting be used in respiration or can be formed into fructose and sucrose and then translocated to other parts of the plant. So the first step is to discharge the plant. Once discharging has taken place, the plant is then exposed to sunlight for a period of 4 to 6 hours, also making sure that all the other factors that are necessary for photosynthesis, like water, temperature, carbon dioxide, are present and optimum. Once that has taken place, then we should expect that photosynthesis will have occurred in the plant, and the excess glucose will be converted into starch. Now, the following are the steps in testing for starch in a leaf. The first one, you obtain the leaf from the plant that has been exposed to sunlight for a period of about 4 hours. Then place it in boiling water. You place it in boiling water. This is to kill the protoplasm, that is to kill the living part, and also stop the process of photosynthesis. In addition, placement in boiling water also helps to rupture the starch grains. The starch granules are ruptured, and if that will make it easy to test for their presence. Then, in the next step, the leaf is placed in either a beaker or a boiling tube containing ethanol. Ethanol is an organic solvent, one that is commonly used to extract chlorophyll molecules from the leaf. After placing the leaf in a beaker or boiling tube containing ethanol, the beaker with the ethanol and the leaf is then transferred to a larger beaker containing hot water. So the boiling tube or the beaker containing the leaf and the ethanol is placed in a water bath, because you do not heat ethanol directly because it is highly inflammable. So we heat the ethanol indirectly in a hot water bath. The heating lasts for about 5 minutes, and during this time chlorophyll molecules will be extracted from the leaf. Now the extraction is important because when testing for starch, the presence of chlorophyll molecules would obscure a positive test. So it is important that you remove the coloring pigment, that is the chlorophyll molecules, from the leaf. After about 5 minutes of heating ethanol, the leaf is then placed in a petri dish containing warm water, or it can also be placed in another beaker containing warm water, preferably warm water. This is to remove residual chlorophyll molecules and also ethanol. And warm water also prevents the leaf from drying and becoming hard and brittle. So after washing in warm water, then it is transferred into a beaker, into a petri dish, or it can be spread out on a white tile. Then you pour, you flood the leaf. You apply as much iodine solution as possible over the leaf, and if it turns to blue-black, then that will show that starch is present, and therefore can conclude that during the time that the plant and the leaf were exposed to sunlight, photosynthesis took place, carbohydrates were produced, and the excess converted into starch for storage. So that way we can conclude that photosynthesis has occurred due to the presence of starch in the leaf.